Hello everyone, I'm Sachin Talathi and I'm here to present the work that my colleague Andre Golard and I did on ultrasound for gaze estimation. While the idea for ultrasound for eye tracking has been proposed before, to the best of our knowledge, this is the first empirical demonstration. First, I want to present some background on ultrasound as it is related to a couple of questions we have been getting as we worked on this project. We are dealing with airborne ultrasound, not the diagnostic contact ultrasound most people are familiar with. Based on the difference in impedance between air and biological tissue, 99% of airborne ultrasound signal will be reflected. In addition, we want to note that we use ultrasound in the megahertz range. This shows strong attenuation in air. We will get back to this later in the talk. Finally, we also want to note that the sound pressure levels produced in our system are safe by several order of magnitude. We did several rounds of modeling before deciding to embark on the design and testing of hardware. In the first round of modeling study, we wanted to convince ourselves that we could, in theory, estimate the gaze angles of a sphere-on-sphere model eye using one source transducer and a handful, in this case 4 receivers. In the second round, we looked at the ability to estimate gaze using an omnidirectional as well as a directional transducer. We found that directional transducers allow for better discrimination of gaze angles. This observation played a key role in our choice of transducers for the hardware platform. We chose capacitive micromachined ultrasonic transducers, CMUTs in short. As you can see from the figure in the left, these are made of an array of individual cells. All cells are connected to the same electrode and counter electrode. Thus, they operate as a fixed phased array. We sourced units with a center frequency of 1.74 MHz. We used the test bench in the middle to verify our ability to measure distances and to estimate signal decay. The target is mounted on the linear translation stage. We use a test bench on the right to measure the emission cone of transducers. The transmit CMUT is mounted on a rotation stage. We used the third and the final test bench shown on the left to gather data for gaze estimation. A sphere-on-sphere model eye is mounted on a motorized goniometer, which is itself mounted on a 3D linear translation stage. Rather than using multiple transmit and receive transducers, we used a pair mounted on rotating stages. This allowed us to mimic all positions of the transducers around a circle. In order to take occlusion into account, we printed a partial face using soft rubber. This piece is mounted against the model eye and allows it to rotate freely. In our experiments, we fixed the receiver and rotated the transmitter along a 180-degree arc in 10 degrees steps. Movements of the model eye and transmitter are automated. Here is an example of recorded raw time trace of ultrasound sensor signal. The top row shows an example of time trace recorded at the receiver ultrasound CMUT sensor in response to a single burst of test signal from the transmit ultrasound CMUT sensor. The middle row shows averaged signal computed from the response to a set of 10 bursts of test signals. Finally, the last row shows the squared filtered response signal out of a Butterworth filter. The red line indicates the time period of time to peak signal detection. Time to peak and peak amplitude signals are then fed into our machine learning algorithms. These consist of gradient boosted regression trees which combine the input of several weak learners into a powerful estimator. We used a pair of trees, one for vertical gaze and one for horizontal. The figure on the left shows signal decay as function of the distance between the transducers and the target in our first test bench. Please note that the total travel distance is twice this value. In a typical glasses setup, the distance traveled from one transducer to the eye and to another transducer on the opposite side would be on the order of 5 cm. The figure on the right shows signal amplitude as function of rotation angle in our second test bench. As you can see, the signal is directional as expected for phased array. We used our third test bench to generate data for different gaze angles and transducer locations. The data was averaged, filtered, and peak time signal and amplitude signal were extracted. We trained a gradient boosted regression tree model on these data samples, performing a 5-fold cross-validation study. The model performance is reported using an adjusted R-squared score and the gaze RMSE error in degrees. We obtained gaze RMSE error of 1.021 degrees and mean adjusted R-squared score of 89.92% with a standard deviation of 4.95%, which suggests that almost 90% of the data fit the regression model. This analysis offers an empirical evidence for our claim that ultrasound sensors can be used for gaze estimation in the presence of occlusions. In figure A and B, we show feature importance for the GBRT tree models trained to estimate the model eye gaze coordinates for the horizontal and the vertical gaze. We see that the top two features for both horizontal and vertical gaze GBRT model are time to peak signal, which is then followed by an amplitude feature. It has been our observation that while the time to flight component of ultrasound signal contains dominant information to estimate gaze, 95% contribution to the regression score, the amplitude signal is also an important contributor for the GBRT model to produce an adjusted R-square score close to 90%. In figure C, we show the mean RMSE error across all cross-validation folds for the GBRT model. The error is biased towards the lower half of the vertical gaze, which is primarily resulting from the occlusions. In conclusion, our study offers the first experimental demonstration for the use of ultrasound sensors for gaze estimation. We showed that ultrasound transducers can effectively produce signals useful to resolve eye gaze within the range tested, plus minus 5 degrees in both up, down, and left-right directions. Our GPRT models show that both amplitude and the time of flight contribute to our ability to estimate gaze. This is a new finding as previous modeling work dealt with time of flight signals only. Two factors contribute to amplitude: attenuation and the incidence angle of incoming sound. We show that attenuation plays a role in our ability to estimate gaze, which would favor the use of high-frequency transducers. For this proof of concept, we averaged traces for an effective acquisition rate of 200 Hz. In future work, we will investigate the use of ultrasound in tracking fast eye motion. Specifically, we plan on using a fast-moving model eye coupled with multiple receivers operating at 2 kHz. The GBRT models will be adapted so we can test the potential for using ultrasound for fast eye tracking to resolve saccades. This concludes our presentation. Thank you for your attention.